Okay. Our last speaker, last but not least, is Professor Jiang, and he was awarded a PhD from the University of Queensland, which is in the north of Australia, back in 2011. He also has a Master of Applied Science in Environmental Microbiology from Lincoln University in New Zealand. That's our cousins just across the ditch from us in Australia. And his research interests include wastewater treatment, wastewater microbiology, wastewater-based epidemiology and microbiologically influenced concrete corrosion. And he'll be talking today about microbial corrosion of concrete. Yep.
Okay, thank you, Scott, for the introduction. Good day to everyone, no matter where you are. So today I will be talking about concrete corrosion. So my background, as you may have already heard from Scott, it's mainly on wastewater aspect, which is a big part of environmental engineering. So for my research on MIC for concrete is mainly for the wastewater collection system, which or you know as sewer systems. So the sewer system is actually a big part of the overall wastewater infrastructure, which including the collection, treatment, disposal systems. But sewer accounts for 70% of that. So it's a massive kind of system. In terms of the wastewater infrastructure, it has usually been built for a very long service life. So most of our wastewater infrastructure has been in there for very long. So that means like for the United States, they need $271 billion to basically rehabilitate their infrastructure for the next 25 years. So that's a huge problem there. So why we have this kind of a problem? This is because in the wastewater system, in the sewer system, so here I'm showing a gravity sewer system. So you will have water phase, you will have a gas phase, and then surrounding that, it's a concrete, it's a solid phase. So for this system, we normally in the past we treat it as a physical or chemical system, but actually one of our pioneers in the sewer processes, he said the sewer is not only a simple transport system, it's also a biological reactor. So with more and more studies, we understand there are lots of bacteria in biofilms or in sediments or in suspended form. They are doing all sorts of biological transformation. And a significant part is the sulfate. So sulfate reduction can happen in the wastewater, especially when it's anaerobic condition. And then the H2S can participate into the gas phase. And then on concrete surface, you will have the sulfide oxidizing bacteria, which turn that into sulfuric acid. So in that phase, then the acid will react with the cementitious material. So that leads to the loss of the mass and the loss of the structural strength, etc. So you already heard from our previous two speakers and also from Scott about the roles of the microorganisms. So here for the MIC of concrete, there are some well-known ones. Acetylbacillus, which is a well-known one. That's more or less kind of most widely accepted microorganisms that causing the problem. But also we believe there are heterotrophs, there are fungi, which they're not only working by themselves, they kind of interact with those sulfide oxidizing bacteria. So overall, this can, causing a very severe problems that including economic impacts. So it's a very good topic to work on. So here, because we only got about 10 minutes, so I will give very quick overview about what I have done on this field. because I started as a environmental engineer, so I only started this research about 15 years ago after my PhD. So many 4 aspects. First part is understanding the corrosion of the concrete. So look at the impacts of environmental factors, the concrete composition or the properties. So it's like those kind of 3 elements related to the MSA. And then based on that, I did some work to develop some corrosion-resistant concrete material for the wastewater system. So one is using the nitrate as admixture, look at different cement types. Another work I did with my previous DECRA is for the bioconcrete development using some bacteria or granules which are derived from wastewater, and then I use that in the concrete. So it's kind of a special idea there. For the applied aspects, I have to develop this kind of a biocorrosion chamber, which has been used quite widely in Australia, but also internationally for some large-scale wastewater infrastructure projects. So I'll quickly go through those 4 aspects. So the first and when we start this is we want to understand what roles do those microbes play in the concrete corrosion. So as you can see, we have different types of concrete, and then we put in the wastewater systems. So we have like different wastewater conditions simulating the spray of the wastewater or the flooding of the wastewater. So those are the typical operational conditions in the sewer system. So from that, actually, we can identify the microbial communities under those different conditions, different types of concrete, different hydrogen sulfide concentration, and then different wastewater like inoculation conditions. So from that study, as you can see, the wastewater actually played a big role in shaping the microbial communities. So like the typical or the most well-known ones actually can be reduced by 30% under that condition because of the flooding of the wastewater. But some other bacteria like acidophilum and Acidobacillus caudes, which can be significantly increased by those conditions. So this shows basically the interactions of the wastewater with the microbes, and then the relationship of the microbes with the final corrosion rate, which is shown here. So that basically establishes the link between those different elements. Of course, we also want to look at the environmental conditions, right? How would they affect the MSA of concrete? So this is a long-term study, which I actually worked on all the lab work for nearly, I think, 5 years. It's a very long-term study. So we look at the temperature, different levels of temperature, relative humidity, different levels of gases H2S levels, and then look at different locations in the concrete pipes system. Some are fresh concrete, some pre-corrode, etc. So here we established a suite of comprehensive assessment of the corrosion by surface pH, chemical analysis for the sulfur species, using photogrammetry for the corrosion loss in millimeter per year. We also basically take corrosion products, extracting it, do sequencing. We also develop a special method, which is called sulfide uptake rate, SUR, to quantify the corrosion rate. Of course, in addition to that, we do all the other microscopic analysis, such as SEM, XRD. We also use mineral liberation analysis, etc. Yeah, so we had quite a few publications on this to talk about the different impacts of temperature, humidity, and H2S on the corrosion, not only on the corrosion rate, which is the active corrosion stage, but also how they affect the startup, the initiation of the corrosion. So I won't be able to go through all those results, but just to let you know, we conduct those research. So if you're interested, we can communicate further, or you can check our publications. And then we also had another question when we work on this is, the corrosion, to quantify that, it's very difficult. The corrosion loss using photogrammetry, which is a destructive assessment. So you need to brush away all the corrosion products to expose the intact concrete. So based on that consideration, we think we need to develop a method that can do this kind of a long destructive measurements. So that's why we developed this SUR with one of our previous PhD students. Basically, the idea is when we expose the concrete to H2S gas, they will uptake H2S. Basically, the microorganisms, they will oxidize H2S. So what you will observe is this kind of decreasing profile of the H2S. So with that profile, we can then calculate the consumption rate, which is called H2S uptake rate. From there, we can then back calculate the actual corrosion rate. So that is a kind of a way to quantify the instantaneous corrosion rate without destructing the sample. So then, of course, we also look at so we have this problem. How can we make some like change to the concrete material to make it more resistant to the corrosion? So one big part of my previous research is based on nitrites, on the chemical aspect in wastewater for a different purpose. But we think this may also work in the concrete. So that's why we work on this nitrite admix concrete to look at their mechanical properties, how nitrite affect the concrete mechanical properties, including those very common ones, strengths and other things. We also look at the leaching behavior of nitrite, because nitrite, people have a concern about it as a increase of the nitrogen load in water, but also impacts on the environment itself. We look at the biofilm development as well. So this is a project we worked through a few projects. I think it was a ASA linkage project and some discovery project as well. So we did lab studies, but also did studies in this pilot scale sewer pipes, which is still quite large scale, 1.3 kilometers long. We also did studies in the real sewer system. So this is a collaborative work with Gold Coast City Council. So eventually, I think we got a good results that we demonstrate this nitrite admix concrete can have good corrosion resistance. Another thing, I mean, as a microbiologist, we always look at some solution based on the microorganisms. So one of the project we did is a bioconcrete. So basically, I guess for the conventional bioconcrete, most of us is interested in the so-called MICP, basically the microbial or induced calcite precipitation. So here we start with that in the perspective. So we want basically idea is we want to use SRB and NRB, sulfate and nitrate reducing factor, which we want to induce the MICP, which may induce the crack healing. That's a starting point. So to achieve that, obviously we need to start with our wastewater cultivation work. So we use this UASP reactor, which is normally used for wastewater treatment. So we use that as a substrate to cultivate the granules of SRB and NRB. And then we test the concrete samples. So I think this is the work we did with ANSTO using their neutron analyzer to observe the granules. We cultivate how they have been distributed inside the concrete blocks. And very interestingly, when we conduct the corrosion test, we found this bioconcrete actually has very good corrosion resistance. So that's a kind of a bio solution to this. Just the last aspect I want to talk about is how we inform the real world decisions with our research. So like I said, I provide this kind of a corrosion test for many large sewer projects like the Singapore DTSS project, which is very large pipelines, 80 kilometer long, and also in Auckland, New Zealand as well. Currently, we are still working with Japan, some in different other countries as well. So we still get some crazy ideas, but I guess more is along with all the corrosion researchers' idea. So like Scott mentioned, I also work on the wastewater-based epidemiology. So which is basically a way to track pathogens like viruses such as SARS-CoV-2 or influenza, but other pathogens as well by analyzing the wastewater. So basically, we look into the wastewater, then we know what's happening in the population. So we want to extend the same concept to the corrosion aspect. So whether we can identify any panel of corrosion biomarkers that could be some microorganisms, could be some functional genes, some metabolites and other things related to the concrete corrosion. And then we can use that for the monitoring purpose, for the prediction and many other things. So we can use that to manage or to mitigate the corrosion. So that's something we still want to work on, although currently mostly I'm working on the virus and other things at the moment. Yeah, so I guess on the concrete aspect, concrete will still be used very widely. The research on the MICF concrete is still not done yet, although like we recently published a book on that. I think there are still many unknowns, especially on the microbial ecology aspects with the advancement of the multi-omics approach, the AI, etc. There are lots of things we can do. Okay, yeah, so last is just some acknowledgement to my team and also the industry partners we have worked over the years. All right, happy to answer any questions if you have.